Okay, we're going to cover chapter number 4, the hemispheric specialization. The big question in this chapter will be like, do the differences in the anatomy of the two hemispheres explain their functional differences? Why do split brain patients generally feel unified and no different after surgery, even though their two hemispheres no longer communicate with each other? Do separated hemispheres each have their own sense of self? Which half of the brain decides what gets done and when? But before we proceed, maybe you just need to remember that if this is, for example, this is a cortex, I should do my best to make it look good. Okay, this is cerebral cortex. Okay, and this is the brainstem. And this is the spinal cord. Okay, so if this is like, if this is the right side, and this is the left side, okay, and this is, you know, the middle. So if there is like pyramidal tracks, for example, like coming from the cortex and goes down, go all the way, it always like cross over to the other side at the end of the midbrain for brainstem. So brainstem has 3 pieces, as you know: midbrain, pons, and medulla oblongata. Usually at the lower part of the medulla, they decussate or they crossing to the other side. This is okay. So they come here and go down. So when they cross over, of course, some of the fibers actually just go straight down as a redundancy, but the majority always just cross to the other side. What that means is like your right side of the brain control your left side of the body, and your left side of the brain controlling the right side of the body. Okay, so if you want, for example, to examine the left side of your brain, usually you're going to show stimulus on the right side of the subject that you are testing. Okay, so this is our famous patient, WJ. He has like split brain because he used to have a seizure, and the doctors perform like split the brain. And you know, the two hemispheres connected. So this is like, let's say this is one hemisphere. Okay, sorry, and this is another hemisphere. This is the right side. This is the left side. They actually both connected something called corpus callosum in the middle, called corpus callosum. Okay, so in case of like patient who suffer like epilepsy, okay, so they got something called seizure. Usually the doctors find out if they actually disconnect the both sides of the brain by splitting the corpus callosum, detach the corpus callosum. They found actually that the symptoms actually will be better. Okay, they won't get epilepsy as much as, or the seizure, I'm sorry, as much as they used to. So this is one of those patients who has the split brain. So as you can see here, this is the pattern in the red right here. This is in front of him. This is a pattern he's supposed actually to form using these cubes to put or to form this pattern. So this is actually the shape, the pattern in red on the white card is the shape that the patient is trying to create with the blocks given to him. At A, here in this image, with his right hand, oh, so what control the right hand again? Will be the left hemisphere, this one, this part. So if this is the brain, this part actually controlling the right hand. And here, left hand, this is right hand that's control the left hand, okay? So this is right hand, left hemisphere. He is unable to duplicate the pattern. He's found it really difficult to duplicate the pattern. That means the left side is not able to do this function. But in B, here, with his left hand or the right hemisphere, he is able to perform the task correctly. Okay? So actually the WJ, a former World War II paratrooper, began having seizure after a head injury. A German soldier hit him with the butt of the rifle in 1962. So Gazzaniga, the professor who wrote this book, ran an experiment wherein he asked WJ to press a button when he saw an image. Gazzaniga's team would flash image of letters, light bursts, and other stimuli to WJ left or right field of view. Because fields of view are processed by contralateral hemisphere, flashing image quickly to one side or to the other send the information solely to the intended hemisphere. So when stimuli were delivered to the left hemisphere, WJ showed no impairment. In contrast, when stimuli were delivered to the right hemisphere, WJ reported that he did not perceive any stimuli. Yet his left hand correctly pressed the button when an image appeared on the screen, according to Gazzaniga. So the left and right did not know what the other was doing. It's very amazing and very actually unique actually case. So the hemispheres actually is like anatomical correlates, and there is something called Weda test. The hemisphere of the brain are distinct yet connected. So the medial view revealed the commissures, the large white matter fiber track that connect the hemispheres. Determining which hemisphere is dominant for a given ability. So the Weda test, which is the sodium amylase test, it's actually anesthetized one hemisphere and check for language function. So for example, again, if this is a two hemisphere, let's just draw them together. Okay, so this is right, this is left. For example, I want to test the left hemisphere. Okay, then I'm gonna anesthetize, stop the function of the right hemisphere by injecting this material called sodium amylase. And then, you know, this is actually done in elective epileptic surgery, during elective epileptic surgery. So when you anesthetize the right side, okay, that means the right side is not working for now, or not seeing the stimuli, and then you start to check the function of the left hemisphere. There is another test, the test actually called the dichotic listening. It's actually testing the hearing in both ears. It reports more digits heard by the dominant half. And the functional brain imaging, of course, and PET used to see which half is active when doing a language test. The asymmetries, they found that actually both hemisphere, the right and the left, they're not really symmetrical exactly, as you can see in the image right here. So the view looking at the inferior surface of the brain, as you can see here, note that the left hemisphere appears on the right side of the image. Okay. In this computer-generated reconstruction, the anatomical asymmetry have been exaggerated. It's exaggerated a little bit to show you that there is actually they are not really 100% symmetrical. Okay. The corpus callosum on the left, this is the one we're talking about. This is a corpus callosum, this part here, nice part. This is a corpus callosum. This actually would connect the right hemisphere to the left hemisphere. So when you actually split the brain, you just detach this, or you cut it, you split it, so right and left cannot communicate, so the seizure will be less. This is a sagittal view of the left hemisphere of post-mortem brain. The corpus callosum is dense fiber tract located below the fold of the cortex, as you can see here clearly, this nice color here. And the anterior portion is the genome. So this is the genome. This one here, this is the anterior one. And the middle portion is the body itself, which has this nice word corpus callosum. And the posterior portion is the splenium. The last one, the posterior. This one, splenium. The structural asymmetry is enlargement of the planum temporale in the left hemisphere. So they're not really, as you can see from here, they're not really symmetrical 100%. And this is a corpus callosum one more time, here, right here, this nice structure here, which connects right and left. Some surgeon actually decide to split the brain when they have a seizure or epilepsy. The mid-sagittal view of the right cerebral hemisphere with the corpus callosum labeled. The caudal surface of the coronal section of the brain, roughly through the premotor cortical area. Homotopic callosal fibers in blue, this blue one, so blue will be homotopic. Homotopic mean would be exactly like, for example, if this is neuron A, let me just go. So if this is actually A, so they connect A and A in the other side. So exact type of connection of neuron, okay, called homotopic. Heterotopic mean if there is A for example, and here B and B, so we'll be connecting A and B, etc. Not A and A. So this will be heterotopic. So the caudal surface of the coronal section of the brain, roughly through the premotor cortical area. Homotopic callosal fiber in blue connect corresponding sections of the two hemispheres via the corpus callosum. Heterotopic in green, which can connect A to B, for example, in different part, not in exactly in the same area. So homotopic mean from area A, for example, to area A here. Heterotopic mean connecting from area A maybe to area B far here, okay? Connect corresponding section of the two hemispheres via the corpus callosum. Heterotopic connection in green link different area of the two hemispheres of the brain. In primates, both types of contralateral connections, blue and green, as well as ipsilateral connections, red, start and finish at the same layer of neocortex. The corpus callosum is a bundle fiber that carries signal between the cerebral hemispheres. Okay, so the red actually will be connecting ipsilateral, meaning in the same half. So let's say connecting area A to area C, same side. Homotopic would be area A from right to area A from left. Heterotopic would be from area A and the right to area B in different place in the left side of the cerebral cortex. The structural and functional connectivity here, as you can see, this is non-synchronized firing, as you can see, so they not resemble each other. So these two separate light bars moving in different direction. Okay, so this one direction, this upper direction, and this is in the right This is right visual field, and this is the left visual field. Okay, so this is actually when the corpus callosum is intact in the middle here. See, if it's good, no problem with it, they're gonna just give you the same firing from the right visual field and left visual field, both firing synchronized. But in the severe, when you sever the corpus callosum, when you split it, so the right and the left is separated, the two hemisphere, they give you non-synchronized firing. So when repetitive field 1 and 2 on either side of fixation are stimulated by two separate light bars moving in different direction, as indicated by the arrow. This is going down here, this is going up. The firing rates of the two cells are not correlated. In B, a long light bar is spanning both fields. Here, in animal with an intact corpus callosum, cells with spatially separated receptive fields fire synchronously when they are stimulated by a common object, such as long light bar spanning both sides in both fields. C, in animals whose corpus callosum has been severed, synchrony is rarely observed. This is a very nice image. Actually, just you need to know that the left, of course, the left hemisphere is well known for what? Language dominance. And here you can see there is actually cellular asymmetry between left hemisphere and right hemisphere. So just by comparing these two sides, which neuron actually is bigger? This one is this side here. This, or this. You're gonna find that actually the left hemisphere is larger. This is actually the layer 3 pyramidal cell asymmetry. This visual examination reveals a subtle difference in the sizes of the largest group of the layer 3 pyramidal cells, stained here with the acetylcholinesterase. You don't have to know the name of the stain. So in the left hemisphere, they are larger than the right hemisphere. So you find this here larger, the cells see larger than the right hemisphere here. This is right, this is left. And the left hemisphere is better in recognizing the self. And the right hemisphere is better at recognizing a familiar others. The proportion of yes response here, the proportion of yes responses to recognition judgment and plotted on the y-axis as a function of the percentage of individual contained in the image and the cerebral hemisphere to which the image was presented. So this is two different actually split brain patient. This is the JW we'll talk about, and this is the MG in the image. So they show the image to the patient. So they were able about like they show their own image, and this is actually show them familiar other. They found that the left hemisphere is better in recognizing self. So if you show like JW his image, he will know it. His left hemisphere will know it, will respond much better. But if you show like familiar others other than JW, his response will be better in the right hemisphere. So bottom line is that the left hemisphere is better at recognizing the self, and the right hemisphere is better at recognizing a familiar others. As you can see here, this is the yes. If they know if the picture, actually they ask him if this is your picture or not, he's gonna recognize it. So percentage actually is higher. See here, this is the left hemisphere. and this is the right hemisphere in blue. For the other patient, for MG, this is the blue, or the right, and this is the left. Voluntary versus spontaneous expression. This is for the facial expression. So this is a motor cortex, and this is a direct pathway or fibers that controlling the right side of the face, and this is the indirect fiber. Still crossing to the other side and controlling the right one. So indirect fibers will be the reticular formation here, and this is the facial nucleus, and this is the facial nerve to the left side. And here this is the caudate nucleus. This is, of course, you know that the basal ganglia has like 3 nucleus, very famous: caudate, putamen, and globus pallidus. This is the motor cortex, of course. This is a caudate nucleus, putamen, globus pallidus, and this goes to, now this goes to the right side, and this one goes directly to the left side. This will be the facial nerve, and this will be the facial nerve. So in the left side, the voluntary expression that can voluntary expressions that can signal intention have their own cortical network in human. On the right. And the neural network for spontaneous expression involve older brain circuits and appear to be the same as those in chimpanzee. The location of the section that has been overlaid into each face. This actually patient with a complete corpus callosum colostomy. So there is not even one single piece here for the the entire corpus callosum was removed. See, everything. And this one, just we left a little bit of piece of the corpus callosum still there. So they removed this part and they left this whitish color here. A complete corpus callosotomy, I'm sorry. So this MRI shows a sagittal view of a brain in which the corpus callosum has been entirely sectioned. The arrow indicates the region of resection. This is a mid-sagittal cut would sever all of the commissure connections. The hemispheres are capable of functioning independently. There is no connection. But this one here, when you leave a little bit of the piece, shows that the splenium arrow, this is the splenium, was spared in the split brain procedure performed on this patient. As a result, visual information could still be transferred between the cerebral hemispheres. So by just leaving the splenium, this little piece, the visual information could still be transferred between the cerebral hemispheres. Does attention actually differ in a split brain patient? Actually, a split brain patient can use either hemisphere to direct attention to a position in either the left or the right side.